Rick Page, if I by chance haven't met you already, it's really my pleasure to kind of kick things off, not just for today, but for the week in terms of Research Celebration Week. Um, the Just to mention, and Kate Tracy, Senior Associate Dean for Research, Kate Tracy, is going to tell you more about the activities coming up this week. I just want to express my gratitude to Kate for reimagining Research Celebration Week. We've got activities all 5 days this week. It's really exciting. Kicking off actually this morning with a breakfast for our graduate students, going on through Friday in the Stetson lecture. So before we get things going, I just want to acknowledge Kate, and let's give her a round of applause. I also just want to acknowledge that this lecture is a really big deal. The Research Laureate is as prestigious an honor as we have in terms of acknowledging awardees from our own Larner College of Medicine. You're going to hear more about our speakers, so I won't embarrass Dan yet and let other people do that. But it's really a pleasure to sit back and listen to him in his talk today. I will also acknowledge that Ralph Budd is here, and he shared with me he was the first Research Laureate lecturer back 30 years ago, was it, Ralph? Something like that. Yeah. So that we had slides we put in the projected. No, but Ralph, thanks for being here. Other awardees that might have received before, please accept my apology for not naming you by name. But I do want to thank everybody for being here. This is, with everything going on, including proclamations Friday night about that could affect researchers, this is a time, this is a week to celebrate our core values and our core missions. And there's no better way to do that than to acknowledge one of our own. Next up to the lectern is Dean Kate Tracy. Please join me again in giving her a round of applause.
Thank you to Dean Page for that fine introduction, and thank you to all of you for making time to be here today. And Bruce, do we have a count on how many Zoomers we have? So shout out to the folks joining us remotely. 20, whoo! So welcome to the kickoff for Research Celebration Week. It's, we've been doing Research Celebration for 9 years now. Laureate went on, I think, a little bit longer than that. And I'm, as Dean Page said, so excited to be able to put the spotlight on all the great work that we do in the Larner College of Medicine every day. We did kick off this morning with a breakfast event with our graduate students. Dan is formally kicking off Research Celebration Week with his talk as the Research Laureate. Tomorrow, we have two special events. One is the Biolabs ribbon cutting at the Colchester Research Facility, which is at 11:30 tomorrow morning, followed by tomorrow afternoon, our Learner poster exhibition, which will run from the Hale Gallery all the way up to here with lots of posters on display. So stop in there as well and see some of the great work being done by our learners. Wednesday, our signature event is the TED Style Talk Research Rally, and some of you in the crowd will be stepping up to tell your stories in 7 minutes or less Wednesday evening. And that's my personal favorite event of the week, although I'm probably not supposed to have favorites. Thursday, we have the awards ceremony followed by my research address, and then we cap off the week with a Stetson lecture on Friday when Dr. Stefan Lindert will be here from UCLA to give the Stetson lecture. So for me, it's a great week. It always makes me feel excited about the college, about the great work that we're doing here, and a reminder that we are among the noblest of professions as researchers, and now more than ever, we need to celebrate that. So without more commentary, I'm going to move toward introducing our Research Laureate speaker, a scientist whose work has not only advanced understanding of lung biology, but has also shaped the future of regenerative medicine. Your friend and mine, Dr. Dan Weiss, to my right here. Dan has devoted his career to one of the most complex and urgent challenges in medicine, repairing and regenerating lungs after injury. His pioneering research in gene and cell therapy for lung diseases has opened new doors for treatment, offering hopes to millions affected with respiratory conditions. Dan exemplifies the spirit of the Larner College of Medicine and Translational Science, bridging discovery and clinical application with rigor, creativity, which we're going to get a little bit of in a minute, and compassion. He is the visionary behind the biennial conference, Stem Cells and Cell Therapies in Lung Biology and Diseases, that began at the University of Vermont. The conference has just celebrated its 20th year, has become a cornerstone in the field, recognized by the NIH, the FDA, and leading respiratory foundations as a vital forum for collaboration and innovation. An interesting sidebar, I actually got to know Dan before I even arrived here to be your Dean of Research when I was still at the University of Maryland, because I got the offer, I accepted it, and in my email box appeared an email from Dan saying, hey, I'm going to be hosting this conference next summer, fully 7 months away, not even in Vermont yet. Would you mind coming by? I'm like, sign me up, I'm there. So I got to know Dan before I was even on site, because that's just how he shares his enthusiasm and promotes the great work that he does. For more than 30 years, Dan's work has been continuously supported by an array of sponsors, including the NIH, the NSF, the Department of Defense, and numerous nonprofit and industry partners. His lab is currently exploring 3 transformative areas: bioengineering, functional lung tissue, ex vivo, immunomodulating lung inflammation using mesenchymal stromal cells, you can tell I'm not a basic scientist, and developing cell therapy-based strategies for lung disease. Now, normally this is the part where I would invite Dan to the podium to give his talk, but we have the option of longtime collaborator, colleague, and friend, and Department Chair Ann Dixon would like to also make some remarks. So I'm going to invite Ann to come to the podium before we bring Dan up. Ann?
Thanks so very much, Dean Tracy. It has been my amazing privilege to know Dan for about the last 30 years. So about 30 years ago, we were trainees together in the same lab in Seattle. A lot less gray hair. I think Dan actually had hair. And what stood out about Dan then is what has stood out about Dan for his entire career. It is his boundless enthusiasm for science. It's his ability to think outside the box and create a new vision. Back then, he was fostering junior people's careers, and he has done that throughout his career. Dan has grown into an extraordinary leader in pulmonary medicine. His work in the lab has been groundbreaking. He also performed the first ever clinical trial in lung disease and stem cells. But I think his proudest moments have been for his mentees. He has been an extraordinary mentor to people who have gone on to have extraordinary careers in their own right. And Dan, it's just been a tremendous privilege to watch you over the last 30 years.
Right. Thank you, Ann. Thank you, Dean Tracy. Thank you, Dean Page. It is such an honor and a privilege to be standing here amongst all these friends and colleagues, and for all my friends, family on Zoom. Again, I'm very grateful. What I want to do today is share a life in science and the fun it's been and the decision making, and most importantly, the people along the way, because that's what this is all about in terms of shaping, again, a life and a career in science. So fixing broken lungs, it's a pretty daunting task. So just to get going in terms of disclosures, I've been very fortunate to have had funding from a wide variety of sources and the like. And more recently, I've been turned into a business person by some people in this room. And a bunch of patents. I actually have 2 companies now that we're trying to make work. But here's the overview, right? Here's a broken lung, all right? It's a nasty looking emphysematous lung, and we want to get it back to a normal lung, both in terms of structure and function. And so we look at means of doing structural repair, and we look at means of doing functional repair as well. And this is tough, you know, it's not an easy thing to do. But why do we do this? And the unfortunate truth is that most lung diseases are still incurable. There is no cure for asthma. There's treatments, but there's no cure. There's no cure for pulmonary fibrosis. There's no cure yet for cystic fibrosis, et cetera. And in parallel, critical illnesses still have, many of them, high mortality. So what we want to try and do is figure out how we can take clever ideas and match them. So in terms of functional repair for acute, high-intensity, transient inflammatory conditions, things like the acute respiratory distress syndrome, COVID, sepsis, structural and functional repair for some of the chronic diseases that you have listed here, adult and pediatric, with respect to bronchopulmonary dysplasia. And then you've got the chronic fibrotic and destructive diseases that are just so difficult to try and deal with, if you will. So the way I look at this is regenerative medicine. And the way I look at regenerative medicine is that, yes, that's a mouse with a human ear growing out of its back. And so the way I look at regenerative medicine is that there are three ways to think about it. The first is that you can stimulate body's own repair mechanisms to heal previously irreparable tissues or organs. So if you got a cut in your skin is generally going to heal. If you get a cut in your brain, so to speak, it doesn't heal as well. And so if we can figure out how to make that happen, this is one of the tenets, if you will. Another way of thinking about it is to, if you can't do that, can you grow new tissues? Can you grow organs on the bench side or the bench top and the like? And this is actually out there for quote unquote simpler tissues, if you will. Skin, bone, cartilage. Now, I'm not an orthopedist, so be very careful in how I say this, of course, but we haven't been able to do this yet for liver, brain, kidney, et cetera. The other way of thinking about this is development of organ tissue adjunct devices. So if your heart isn't working, there's a left ventricular assist device. If your kidney isn't working, there's a dialysis machine. And hopefully there will be portable dialysis machines as well. And so this is a way that I've kind of framed my view on trying to fix broken lungs, if you will. Now for fun, some of you know this answer, some of you don't, but who was the first regenerative medicine scientist? Any ideas? She, right, wrote a genre founding book in the 1800s. You all know this. Of course, we're just playing catch up, all right? So this is the type of thing where a lot of the science fiction writers had it pegged from years and years ago. So when you look at the spectrum of things that are out there for fixing broken organs, but broken lungs, there's some really impressive and some really fun technology. So going around the horn here, going from stem cell work with a whole variety of stem cells, imaging approaches, cell therapies, organ on a chip, organ on a slice, and some engineering approaches that we'll get to a little bit later. And so there's a good amount to pick and choose from. So how do you do that? So I want to go back to the beginning, all right? So this cute little kid, right, was growing up, and I was always fascinated with science, all right? Always in the outdoors, looking under rocks and, you know, what was there and things like that. And then one of the first major influences that helped me think about a career was this guy. Everybody remember this guy? Anybody remember this guy? The undersea world of Jacques Cousteau. I was going to be a marine biologist. All my buddies were going to be marine biologists back then. And so that didn't happen, but I'm very proud of my daughter, who is a marine biologist and who I live vicariously through. And here she is playing with a tiger shark, you know, and all that. So, but I went to college thinking about marine biology. And this is, again, where the importance of mentorship and the people you meet along the way help shape your life. And so Klaus Beinbach was a renal physiologist in the Cornell Veterinary College. And I worked with him, did a senior undergraduate research project and the like, right? And so he got me thinking about, you know, what are you going to do with your life? And you have this range of interests that probably you should be going to medical school. I hadn't really thought about medical school. I was going to be a scientist. But I ended up doing that. And so I went to Mount Sinai and I did the medical scientist training program. Now, some of you will remember we tried to get that going here years ago. And it's a great program, particularly because it's a full scholarship. And but we didn't have the bandwidth here. That's all fine. But back then, endocrinology, it was all about endocrinology because in the 80s, dating myself, endocrinology was where a lot of the basic science in medicine was taking place, all right? And so my first paper back here with my thesis mentor, Early Ogerpity, all about estrogens and anti-estrogens and the like. But then along the way, I've met more people who helped shape my career. Terry Crowich, the head of the MSTP program, Sandy Mazer, another physiologist there. And they got me thinking even more broadly. But I went on to internship and residency at Michigan, still endocrinology. It's going to be endocrinology. So I actually went for a research project down to the Pima Indian Reservation in Arizona and work with hyperinsulinemia and polycystic ovarian disease and the like under the auspice of a very nice gentleman, Bill Herman. But then it was second year during residency when I did my first ICU rotation. And that was it. I saw the light and that was no turning back. So two incredibly influential people, Cyril Grum and Bob Citrin, pulmonary critical care intensivist at Michigan, just opened my eyes. And so that's what I did. I took a year off though between residency and fellowship. Worked as a ER doc in rural Michigan. Did things I will never do again. I don't even want to think about in terms of little babies and trauma, all this stuff. It was fun, but an experience in life. So I went on to University of Washington and ended up fellowship there and was an attending faculty at Fred Hutchinson Cancer Research Center. And then there are a series of extraordinary individuals listed here: Len Hudson, Dave Pearson, Tom Martin, who unfortunately passed away last week, Dave Mattis, Denny Liggett, and then Joan Clark, who was my and Ann's research mentor during our postdoctoral training and fellowship. And we were going to cure cystic fibrosis. This was the early 90s, and the gene for CF, the cystic fibrosis transmembrane conductance regulator, right, the CFTR gene, was just discovered. And we were going to cure CF in 6 months with gene therapy. And that didn't happen, right? But we learned an awful lot. And so one of the things I got involved in was a lot of fun, was liquid ventilation. Does anybody remember liquid ventilation? Thanks, Ann. Anybody else besides Dr. Dixon? All right, so liquid ventilation was the idea where you have actually a series of compounds called perfluorochemicals. And these are essentially hydrocarbons in which the hydrogen atoms have been replaced by fluorine atoms. And you end up with a clear, colorless, odorless liquid. And here's a rat submerged in a perfluorochemical and is breathing, because the gas solubility for oxygen is about 100 times that than in water. And so we came up, I had this crazy idea that we could use perfluorochemical liquids to improve recombinant gene therapy. So recombinant viral vectors put down the trachea here. And the gene of interest is marked in blue. So this is a rat lung, and these are airways, and all the blue is where we put a recombinant vector down the trachea, the gene turned on, and there you go. And it's not bad. But we made it so much better if you chase the recombinant virus with a perfluorochemical. And it was pretty wild. The rats were able to breathe, and the perfluorochemicals are volatile, so they evaporated and leaked off. But anyway, it didn't work. I mean, technically it worked, but we didn't cure cystic fibrosis. So then I got recruited here in 2001 and just had the privilege of joining an extraordinary group of individuals, all of whom you know, and the like. Some of you, Jerry Davis, retired a few years back, as did Ted Marcy, but these were and have been my friends and colleagues forever. But I still wanted to cure cystic fibrosis. So we moved over to cell-based therapies. And the idea here was, could you take a stem cell, and in this case, Kate, the mesenchymal stem cell, stromal cell, and get it to turn into a lung cell? And the idea was based on some literature that had just come out in 2001, in which you could do clever experiments, like take a mouse and do a bone marrow transplant or inject bone marrow-derived cells. And if you're even more clever about it, you would label the bone marrow-derived cells with a fluorescence marker, green fluorescence protein, And if you're even smarter about it, you would take male cells and put them into female mice. So you could track by doing fluorescence inside your hybridization for the Y chromosome. And so we did that. And here is a photomicrograph of a mouse lung. Here's the airway. Here's the epithelial lining, the underlying parenchyma. And these two green cells are staining for the cystic fibrosis transmembrane conductance regulator. We had taken wild type cells and put them into CFTR knockout mice. So these cells came from the donor, and the little red dots are the Y chromosome. And so we got cells from the bone marrow injected systemically, injected intravenously to engraft into the airway epithelium. Hey, we cured cystic fibrosis, right? Problem being is that there are only two of them. And doing some quantitation, less than 0.01% of the airway epithelium was replaced this way. So we didn't cure cystic fibrosis, but it just set me on this path to do all these different things. So we still do a lot of work with stem cells. I want to talk to you a little bit about the cell therapy and some of the inventions we've come up with and the like. So first of all, cell therapy. So mesenchymal stromal cells are cells that come out of the bone marrow or can be isolated from the bone marrow, adipose tissue, placental tissue, and the like. And they have a range of extraordinary properties. They secrete lots of different anti-inflammatory mediators and growth factors and the like. And you can put them in vitro culture, so benchtop culture with a range of inflammatory and immune cells, and they will turn off the function of those cells. All right. They also are not initially don't have much immunogenicity, so you can do non-HLA match transfer. I can take my MSCs, inject them into Dr. Bonnie, and there would be no initial immune rejection or response. So that led to a huge amount of clinical work in terms of clinical trials for autoimmune and inflammatory disease, graft versus host, Crohn's, multiple sclerosis, et cetera. And they are approved in Canada, New Zealand, and now in the United States for refractory pediatric graft versus host disease. And European Union for Crohn's disease, anal fistula is related to Crohn's disease. So what about lung? So this is a review, good friend of mine, Mauricio Rojas, and I wrote 12 years ago, that there are over 500, and at this point probably 1,000 publications saying that if you take a preclinical model, a rodent, a pig, what have you, and you induce lung injury of some type, and you can go around the circle here, acute lung injury, COPD, bronchopulmonary dysplasia, et cetera. And in virtually every case, if you inject these MSCs intravenously, or if you put them down the trachea, so a direct airway administration, then you will mitigate whatever your injury endpoints are. And there are a variety of mechanisms. Again, all these soluble mediators, extracellular vesicles, which we won't talk about today, mitochondrial transfer, also indirect influence. So for example, in models of asthma that we had worked on with some very talented postdocs, it may be that the MSCs are going to the draining lymph nodes, altering T cell responsiveness and biology that lead to a mitigation of asthma symptoms. So how does this work clinically? You take the cells, allogeneic or autologous, you culture them up, you put them intravenously or the airway. If you put them intravenously, the first pass, the first circuit through the blood, they end up in the pulmonary capillary system and they stay there. And they don't engraft, they don't turn into lung cells, but they'll stay there for a couple days. And then they react to the inflammatory environment, and then they release whatever portfolio of anti-inflammatory mediators, and then they're cleared out. And so we took this and we were approached by a company, Osiris, and my disclosure, I'm never employee of Osiris, but I helped them run a clinical trial. And as Dr. Tracy alluded to, we did, many of you don't realize this, the first ever stem cell trial for a lung disease here at UVM a long time ago, 10 years ago. But again, multi-center, randomized, double-blind, placebo-controlled, 65 patients, 4 infusions over 4 months. There was no rhyme or reason. There's no right or wrong. This is an empiric trial. And the endpoint primarily was safety because we didn't know. These are people with damaged lungs. It includes damaged pulmonary circulation. So if you injected a stem cell, would that clog up and cause a pulmonary embolism and bad outcomes and the like? Secondary outcomes were efficacy, and we did follow patients for 2 years, so it was a good study. Here's the timeline, here's the consort diagram, who got the study drug, procainamyl, placebo. And so we did 10 patients here, and so I sat through, each patient got 4 infusions. I sat through 40 infusions at the bedside, and it took about 2 hours each, because we didn't know if they were gonna keel over, and die in front of us, and they didn't. So there were no infusional toxicities, no signs of pulmonary embolism, and then no adverse events over a 2-year follow-up. Now, so that was great. This established safety, which is, again, the primary endpoint. The study was underpowered for efficacy, and there was no eureka moment, if you will, in terms of pulmonary outcomes, diffusing capacity, 6-minute walk, et cetera. But we did find a change in circulating C-reactive protein. So if you had elevated CRP at study entry and you got the study drug, you went down significantly and over the course of the 2-year follow-up period. So that was really interesting. Something was going on. But it wasn't until 10 years later that I actually thought about this and said, well, why don't we stratify patients based on their level of baseline CRP? And why is that important? Because it's associated with higher mortality, worse exacerbation, et cetera, COPD. So if we stratified based on just simply above and below a certain limit, it worked, all right? So here you have FEV1, forced vital capacity, 6-minute walk distance, so standard pulmonary function outcomes over the timeline, and you can see study drug improved over an initial transient period in the patients with the baseline elevated CRP, peaked at 120 days, 30 days after the last infusion. And these are both statistically significant and clinically meaningful improvements. So an administration of this type of stem cell actually can give benefit in this patient population. So there's a lot to do still, but it is something worth pursuing. But this led to one of my passions, which is stem cell snake oil. And so you've all seen these pictures, right? Come in, get your CBD and your stem cell therapy. God, pain, go get stem cell therapy. And this is a real problem. And so it's based on the realities that few therapies are standard of care or approved. And you're a patient or you're a family member of a patient. It's a horrible place to be. And none of us hopefully will be in shoes like that. And you're going to look for anything. And so if you're bombarded with these false claims and the like, you can understand where this will go. And this has been a worldwide proliferation of clinics, unproven, untested, potentially dangerous. And of course, different regulatory frameworks allow this to happen. So we fought back. And so this was something hooked up with an investigative reporter, Ken Picard, 7 days, and we had an expose on a clinic that was opening up in Vermont. And we actually approached the medical society, Vermont Medical Society, and had resolutions passed that essentially these are dealing with education. So patients seeking stem cell therapies, safe and evidence-based information and services. So this was an initial step. But we took it a step further. And with a really powerful coalition of individuals and state organizations and the like, we got a bill passed by the Vermont State Senate and House in 2021. And I'm really proud of this because it helps to regulate the stem cell clinics that do open up in Vermont. And it's very simple. It's just saying that you have to prominently display the fact that what you're offering is not approved, all right? It's not proven. And amazingly enough, a lot of the clinics out there did not obtain informed consent about these procedures, which is a keystone of patient care, and particularly for unproven and experimental care. So this was really good. But then we took it nationally. And so I never set out to do this, but I ended up being an expert witness in a couple of trials of some of these nationwide stem cell clinics. So the Lung Institute based in Tampa was offering stem cells for COPD, pulmonary fibrosis, et cetera. They quoted my work wrongly, of course, and so I lost it and became an expert witness for the prosecution. And just last spring, they lost big time, so 9 million. Then there's this amazingly bizarre case of a state representative in Missouri who was an assistant physician, all right? So you go to medical school and then you hang up a shingle. You don't do internship and residency. So that's allowable in Missouri, a couple other places. She was marketing what she called stem cells for emphysema, COPD, and COVID. This came out in the COVID. But what she was offering wasn't even stem cells. It was amniotic fluid that she purchased from the University of Utah. Legitimate. Utah didn't do anything wrong. She said, these are stem cells I'm injecting into and will cure your COVID. So you can see fundamentally how crazy it was and the like too. So she's in jail right now. But but everybody remember this guy? Everybody know who this guy is? So his defense attorney was her defense attorney. So I had the surreal experience of being cross-examined by the shaman's lawyer. And suffice to say, he didn't do his homework and didn't turn out well. So I was happy about that. All right, anyway, so that's cells. But let's go back now to growing new lungs on the benchtop. So make sure we're doing time. Okay, good. So this is, again, regenerative medicine, and I want to grow a new lung on the benchtop. So how do you do this? All right, so you need to take into account that the lung is a very complex structure, function, relation type of organ. And so if you're looking for your minimum requirements, gas exchange, oxygen in, carbon dioxide out, you have a variety of design requirements. And the lung on a microscopic level is really a complex organ. So you have all these different cells going from trachea down to alveoli. And at last count, there are at least 40 different types of cells. So how do you want to do this? Well, you got to start with something. So you start with your erector set. You have a scaffold of some type. And best if you can then add organ-specific cells. Best, even better, if they're from individual patients. So if someone's going to need a lung transplant down the road and there aren't enough lungs to go around, which is part of the problem, if you can tailor make a new organ with a scaffold and their own cells, then you've gotten around a huge problem in terms of organ rejection transplant. It's kind of the ultimate in personalized medicine. So we tried a variety of different things, and it turns out the lung is its own best scaffold. So many of you have heard this before, but you can decellularize organs. So here's a native lung lobe after all the cells have been stripped out. And then we can grow these things, and it's alive. So how do you do this? It's a sequential wash with detergents and other agents, sodium deoxycholate, et cetera. And this is the lung we get from autopsy here. We get all our lungs from autopsy. And you can see native all the way down to the whitish looking cell-free organ. And we can put scopes down the airways and the vasculature. So we preserve the macrostructure. We also preserve the microstructure. So here's a mouse lung that's been decellularized, and you can put a cannula into the trachea. Here's the heart scale bar. So electron micrographs show the various lung regions with preserved architecture. We can perfuse dye in the pulmonary artery. It comes out the pulmonary vein, and we've got all types of cool bioreactors to try and grow the lungs in. But it's a challenge, right? So if you have, as an example, a real live lung that's going to be transplanted, and you have a donor and you have the recipient, how long do you have to keep that lung alive before it goes bad on you? You guys know this. Looking at my pulmonary critical care colleagues here. 8 to 12 hours, right? And so, you know, here we're trying to keep a lung alive for weeks while we're trying to grow new cells, and it's a challenge. So, but we can do really cool things. So with long-term friend and collaborator, Darryl Cotton at Boston University, proof of concept. These are mouse embryonic stem cells from a transgenic mouse in which the surfactant protein, which is a marker of type 2 cells, will glow green, if you will. And so we can isolate those from the mice, and we put them into a decellularized mouse lung photomicrograph, and you see all the pink lattice work. And then we got all these purple nuclei, the cells engrafted, and they re-alveolarized, if you will. And more importantly, they actually took on a morphologic and immunophenotype of a type 1 alveolar cell, so a gas exchange cell. So pretty amazing stuff. So when you're trying to do this, a lot of considerations, right? So you start from your native lung, you decellularize it, and there's not one right way. There are a variety of ways of doing it. Then you want to understand what you have left in terms of the scaffold. And then you want to figure out how to get all those 40 cell types back in. What combination, what growth factors, what order, and the like. So it's pretty daunting. We actually call it the zip code hypothesis, right? You don't want cells that should be in the airway living in the alveoli and vice versa. So you got to figure out how to do that. And then the lung is a dynamic organ as well. So you have to take into account cyclic stretch, right? And mechanotransduction and things of that nature. And then you got to implant it at some point. So even if we can do that with human lungs, and we've been working very closely for years with a company called United Therapeutics, who's a leader in this field, there still won't be enough human lungs to go around. So the answer is pig lungs. Now you all saw last year the 2 individuals down at Bellevue, I think was, who got pig hearts, right? Native pig hearts. And more recently, a couple of pig kidneys. So we've been working through United Therapeutics with the same company, Revivicor, here, that makes a transgenic pig that lacks alpha-galactosylase, which produces alpha-galactosylated proteins, which are the major immunogenic foreign protein recognized by humans and pigs. And so we've done that. And so we can decellularize pig lungs, and we can put human lung cells into them. And we're still doing a lot of work with the United Therapeutics on that. So this is going to happen at some point. It's not going to be the same as the heart and the kidneys because those are intact organs. These are decellularized lungs that have been repopulated with human lung cells, right? So that's the difference. All right, so along the way too, you end up doing things that you didn't think you'd start out to be doing. And so one of the things that we had to figure out was how to do high throughput with these decellularized lungs. If you're doing rodents, mouse lungs are yay big, and you can study a lot of mice for all those permutations. But if you move to the pigs or human lungs, they're big and you can't do high throughput, right? So we ended up figuring out if we cut out little thumb-sized chunks and then we're able to cannulate the airway and the blood vessels, we would have these little baby lungs that we could use to study. But the problem there was that you lost the pleural coating, the outside lining of the lung. And so anything you put into the lung just leaked right out. And so we ended up having to come up with an artificial pleura. And we went through a variety of different materials, none of which worked, and because you need all these design criteria, biocompatible, retained cells, et cetera, things like that. And then we came up working with some colleagues in engineering and came up with the idea of alginate, so seaweed-based alginate. And a lot of you have seen this before, but alginate is used increasingly in food, cosmetics, medical use, et cetera. Safe, non-toxic, and it's also fairly easily chemically modified. So by methacrylating alginate, you give a flexibility to the alginate backbone, so you can see where it would be useful for lung. So we say, this is great, we can use this for our decellularized lungs, but then we realized there was a more immediate need. And again, I didn't start out trying to do this. So these are some gory pictures about injuries that can happen to the lung and where they come from. And in particular, bronchopleural fistulas. So a hole in the lung that doesn't seal up, and you have a continuous air leak. And we can put chest tubes in and drain out the air from the chest, but you haven't fixed the problem. And this affects a fair number of patients with the associated healthcare and associated economic costs every year. So we said, well, why don't we take this artificial pleural material, this alginate, and turn it into a Band-Aid, all right? And so we did that, and this was in the context of what's out there now. Surgical repair has these problems. Glues have been tried and been ineffective, so things like fibrin glue, things like cyanoacrylates, so super glue, just don't work. And there's only one currently approved sealant for the lung called Progel, and it has very limited indications and is contraindicated in a variety of things like pregnancy, kidney disease, allergies, and the cardiothoracic surgeons really don't like it. And like it's like caulk in your bathtub in a sense. So we said, well, why don't we do this? And why don't we be even smarter about it and go back to nature and looking at what's out there that's sticky. And so we went to mussels, all right? And so mussels, the shellfish, have these tendrils on one end that allow them to adhere to rock surfaces and the like. So when you blow up the foot, of all things, the adherent compound or the adherence compound is dopamine. And there's a rapidly expanding tissue sealant use for dopamine. So what we did was we conjugated that methacrylated alginate, flexible alginate with dopamine, and we made a patch. And it works like gangbusters. And so we've done now a number of years with DOD, oops, come back, Department of Defense and NIH support. Here's a photomicrograph of a rat, intubated, anesthetized. Here's the lung. We make a hole in the lung and we put the patch on. And if you just look at pressure volume here, baseline non-injured, injured, air's leaking out, and we seal it. And it works. Easy to manufacture, shelf-stable, ready to use. We're in the middle of a large number of pig studies right now, and it's working great in the pigs. So right now, we're looking actually at an IDE, investigational new device, submission for 2027. We've been very fortunate now to have partnered with the Mayo Clinic, and that brings so many advantages to the table, not the least of which is the Mayo Clinical Trials Network that we'll be able to utilize. And this is one of the two companies I formed to help push this along. So very exciting stuff, but again, I didn't start out to do it. It just shows that, as you're growing up in science and you just have an open mind and just think and be creative, strange things can happen and the like. So, all right, so the last thing I want to leave you with is another, crazy idea. And this gets back to, again, those 3 tenets of regenerative medicine, fixing an irreparable tissue that normally doesn't heal itself well, growing a new organ on the benchtop. Well, how about a device? So when you look at lung devices, they're really not a whole lot out there. The major one is extracorporeal membrane oxygenation, ECMO. And here's ECMO. And it's incredibly as complicated as the picture makes it look. High cost, complexity, limited utility. You have to really have specialists. Now over the many years, we've had spurts of ECMO being done here over in the hospital, and it doesn't last long. And you need, again, like any specialized procedure, continuous use to maintain training and expertise and competency and success. There's another ECMO, I think, push up and going down, and good luck and all that. But it also has complexities. So we said, well, why don't can we build a better mousetrap? And again, a lot of you have seen this, but how do birds breathe? All right, let's go back to nature, all right? So here is a moving picture of a bird lung, okay? So birds breathe in and out like us, but their lungs are alien creatures, all right? They're not like ours, all right? So if you follow the yellow goes into these what are called posterior air sacs, and then air gets pumped in a unidirectional fashion through a static wafer-like unit, which is the bird lung. Blood is pumped perpendicularly, so it's not a counter current like kidney, it's a cross current, okay? And it's 100% efficient in terms of gas exchange. Now, we're pretty good, right? Mammalian breathing is not bad, right? We're all here. But there's air that goes in that doesn't participate in gas exchange. All that air that's in the trachea, the large airways, the medium-sized airways, doesn't get down to the alveoli, doesn't participate in gas exchange. So we have wasted ventilation. It's called dead space. You've got one of the experts here in the world with David on that. And so birds have no dead space. So when you look at examples, fun examples from nature, there's a Himalayan snow goose that can fly over the top of Everett. You've got the migratory birds that fly thousands of miles. twice a year. And yeah, they've got wings, but they've got better lungs than we do. So we said, well, why don't we take advantage of that? So why don't we take bird lungs, decellularize them, and then recellularize them with human lung cells, all right? And so here's chicken lung here that we decellularize. Here's an emu lung. That's what an emu lung looks like. And there's an emu farm in New Hampshire that harvests the emus twice a year that we get the lungs from. And this photomicrograph then shows, where are you? This is a decellularized chicken lung, and all these blue dots are human lung cells, very happily growing in a decellularized chicken lung. So we said, well, that's really cool. Why don't we make a device? And so we made something called the Avian Lung Assist device, all right? And it looks like this. So the idea is you have the recellularized bird lung in some sort of chamber, and they've got all types of circuitry, heat control, pumps, and the like too, and you integrate it into circulation. And it's a gas exchange device, right? It's not gonna replace a lung because it doesn't ventilate, but it's like the left ventricular assist device. It's an adjunct device. And better still, if we could personalize this with patients' own cells and the like. And there are three potential uses for this. The big one is in place or as an alternative to ECMO in the ICU. We have a medium-sized backpack device, which is meant to be a bridge to transplant, or for people who unfortunately end-stage lung disease don't qualify for transplant for whatever reason. And then the ultimate is a little implantable device that goes into circuitry with the existing cardiopulmonary circulation. And this is what the backpack device would look like. So again, we didn't start out doing this, but it was a really fun idea, and it's taken on a life of its own. All right, so the future is bright for this. Regenerative medicine and fixing broken lungs. So structural repair, functional repair. I want to give a shout out that Dr. Tracy had done. We have a conference, this has entered the lexicon as the Vermont Stem Cell Conference and has helped put us on the map as an institution. And we did have our 20th anniversary conference this past July. So all work, no play, no way, all right? So most of you know Sharks in Champlain. I kayak all the time when I can. Play bass. I run a music festival. We've raised over $150,000 in charitable donations over the years. Gil Allen and I have done the penguin plunge for almost 20 years, dragon boat, all types of fun things. So you got to enjoy your life, right? I'm indebted, of course, to my family, my father, mother, brothers, Tony and Mike, grandparents, daughter-in-law, my granddaughter to be coming up. So again, so important. Partners in crime. I put a lot of you up here. There's so many more that belong up on this picture. You know who you are, and you know how important the friendship, comradeship, collegiality has been over all these years. Like mentees, Anne very nicely mentioned mentees. I've had the pleasure of having a lot of people come through my lab, a lot of really smart, good people. And we've had a lot of fun, and there's some individuals who have gone on to extraordinarily successful careers, some of whom are showing up here, Darcy, Fernanda, Sarah, and the like too. And this is what it's all about. This is the fun of doing science, being able to communicate the passion. You know, if you wake up in the morning and you're looking forward to going to work, that's, even if you don't make enough money, you know, that's, I'm kidding. That's what it's all about. I also want to acknowledge some additional mentors I've had over the years. Diane, Darwin, Dame Julia, Zia, and Ivan, who've meant a lot for my personal and career happiness and development. And with that, there's so many people to thank. People in my lab, gone through my lab. We've been fortunate with funding. Also been very fortunate to have many, many, many external collaborators, friends, colleagues, and the like that have been good to me. And then again, family. I had 40 years with my wife, and I miss her a lot. So with that, thank you for your attention, and This is my motto, right? And this is what I hope you all will espouse. So thank you again very much. All right, thank you. Great.
A great way to kick off Research Celebration Week. We do have, I think, about 7, 8, 9 minutes. Do you mind taking a few questions?
No, I talk fast. I don't think so.
Wonderful. Any questions from the audience? I can't tell, Bruce, if we have anybody online asking questions. So if you could take a look at that. Questions, burning questions?
Dr. Bud.
So much. Going back to your stem cell trial, patients.
Yeah.
You saw the C-reactive protein death. So is that an indication that disease to start with, or disease to start with first, it might be below that greater diffusion inflammatory rather than the steroid? I know these stem cells.
Right.
Got that.
So that it's a brilliant question, and I hope you have a couple hours for this one. The working hypothesis. Now, I'm the former chief scientific officer and the president-elect of an organization called the International Society for Cell and Gene Therapy. And one of the things that we're promoting is the fact that cell-based therapy, and in particular mesenchymal stromal cell, the MSC-based cell therapy, is going to work best in a more inflamed environment. All right. And when you look at a lot of diseases, they're not homogeneous. Look at the example we're pursuing particularly is ARDS, acute respiratory distress syndrome. And it's clear it's a spectrum of clinical presentation, a spectrum of inflammatory background, if you will, milieu. And so the clinical trials for MSC and ARDS, COVID ARDS, actually work in aggregate. People don't really appreciate this. There is a cure, if you are, treatment for COVID ARDS, but they didn't work for everybody. So we're doing a project right now where we're taking the 8, 9 clinical trials that were published over the past couple of years and trying to get individual patient data from them. And the hypothesis is that the inflammatory state of the patient at study outset is going to predict outcome in response to the therapy. So a little bit long-winded, but the more inflamed you are, the more likely you're going to respond to MSC-based cell therapy, which is what that CRP data showed.
Great. Other questions?
Dr. Bonnie,
That was totally awesome. Thank you.
you saw your picture up there.
Oh, yeah, totally awesome. Yeah. So I never knew your first paper was about estrogens and anti-estrogens. And, the other thing of it is that, everybody knows that lung disease is different in men and women. And so I want to encourage you to go back to your first paper and consider the effects of progestins and estrogens on lung biology and on the success of transplantations.
I left that behind a long time ago. No, it's of course it's a good question, and there are smart people working on sex differences in lung disease and response to therapies and the like too. It's just not me. Yes.
Margaret?
Thank you, Dan. That was a great talk. I really enjoyed it. My question is about CF. Have you given up on your CF cure dream, or are you going to leave it to CRISPR?
No. So we're still working with cell engraftment, actually, not directly. The colleagues I mentioned, Carol Cotton and the like at Boston University, have actually been able to get much better engraftment by putting not the bone marrow derived stem cells, but lung or induced pluripotent stem cell, iPSC derived lung cells down the trachea. So we're helping them with the fluorochemical liquids and the liquid ventilation. So no, not at all. And, Matt Wargo and I circle around, CF all the time and the like. And, so yes, absolutely, but not hands-on here right now.
Other questions? Any online, Bruce?
This is from the Wagner Lab in Montreal. Why do you think the perfluorochemicals improve the efficiency of gene therapy? And is there any other materials that could work given the challenges with PFCs?
Thank you, Bruce.
So that was probably Darcy Wagner, who is a former trainee who most of you know, who's done phenomenally well. She is now the Canadian Excellence in Research Chair of Lung Regenerative Medicine at McGill University. It's a really prestigious position. So there really is no downside to the PFCs. To get a bit more into the weeds, PFC liquids and aqueous solutions are immiscible. It's like oil and water. They will not mix. So the idea being is you take your gene transfer vector or your cell, which you're doing now, as Margaret was asking about, pop that down the trachea in saline or something, and then you chase it with a perfluorochemical liquid, and essentially it distributes it more evenly and more effectively throughout the entire lung. And then the PFC evaporates and the animal breathes it out. There is some data we developed that you could also transiently open up tight junctions between the epithelial cells, which is important for viral vectors that would need to go to the basolateral surface of a air epithelial cell to find its binding site, but that's less relevant for the cells. So it's really simplistically mass action, pushing the stuff in more effectively and more efficiently.
Last question. All right, join me in
All right,
giving
thank you
Dan
again.
another round of applause for a great talk.
Oh, thank you again. Yes, it's fine.